Hello, good morning everyone. We know that we are going through a severe health crisis because of this COVID-19 pandemic, and in India specifically the second wave of infection is turning out to be very disastrous, and that is resulted because we had been reluctant to follow the COVID-19 guidelines from the first wave. That's why we are suffering now. That's the major reason, in fact. So the only purpose is to teach that be careful, follow all the guidelines, and I wish this wave is the last one and hope we learn the lesson of being careful in future also. So the experts believe that to end this pandemic is to have at least one effective vaccine. So the vaccine is the only way to end this pandemic, otherwise it will be very difficult. New mutants will keep coming again and again, and that's why the entire world is being vaccinated today. And even everyone, including from the people from science background or not so, they are aware about the term vaccine. So in today's lecture, we will talk about scientific basis of vaccine development, how the vaccines are produced, and also in context to the three vaccines available in India, Covishield, Covaxin and Sputnik V. So these are three vaccines currently available, and many people ask which is the best one to take. So that answer to that question also will surface during this lecture. But before that, we need to know about the immune system. Our body has an immune system, a system which fights the foreign invaders, the foreign pathogens, for example, bacteria, viruses, fungi, and so on. Because in outside environment, microbes are always there, they are surrounding. So whenever any microbes enter into the body, they are set of cells which get activated and they eliminate that infectious particle. So that is part of immune system, and immunity has been divided into two parts. It's called innate immunity and the adaptive immunity. So when just infection has occurred to the body, there are some cells which engulf that pathogen and eliminate within few hours. So that's a part of innate immunity which immediately occurs after infection, and many times even you are not aware that your body had just fought a battle with foreign pathogen. But many times, like in case of coronavirus, this innate immunity is not sufficient. The cells of innate immunity, neutrophil, natural killer, mast cell and so on, they are not able to completely eliminate virus. So in that case, a second stage of immunity will develop, and that is some cells like macrophages and dendritic cells. When they get infected, some piece of the virus they present on their surface. That's called antigen presentation in immunology. So that antigen presentation in turn activate T lymphocytes, the another very important cell, crucial cell of immune system. T lymphocytes, these T lymphocytes kill that cell which has just got infected by the virus. And because by killing that cell, even virus inside also get killed. Virus cannot survive outside the cell, it cannot replicate outside the cell. So if the host cell is killed, virus also will be eliminated automatically, and same time these T cell also activate B cells, and these are the B cell which produce antibodies against that virus particle. Antibodies are they are very highly specific protein molecules which bind to the pathogen against what they have been developed and neutralize that. And this process of development of antibody by B cell, that's part of humoral cell response, and that takes in fact days, in fact a few weeks to develop antibodies against an invader, any pathogenic bacteria. Whereas innate immunity, it works within few hours of infection. The same thing happens in case of coronavirus infection also. Coronavirus is a spherical particle. We know that on the surface you have many protein molecules which are coming out on the surface. They are called spike glycoprotein or surface glycoproteins. And when the infection occurs, these surface protein bind to some receptor on the host cell, ACE2 receptor in this case of coronavirus, So after binding to the receptor, the virus enters to the host cell, and after entering to the host cell is capture the machinery of the cell, metabolic machinery, and use it for its own welfare, for producing more and more virus particles. So that's how it replicates inside the host cell. Eventually host cell dies, and that produce more and more virus particles which can further infect more and more cells. Then these cells which are infected by the virus, they work as antigen presenting cell also. Antigen presenting cell means these cells show some piece of the virus on the surface, mostly the protein which are present on the surface of the virus, for example glycoprotein, the surface glycoprotein. So piece of those surface glycoprotein is shown on the surface, that's called antigen presentation. And these antigen presenting cell, they activate one set of T lymphocytes called T helper cells. These T helper cells in turn activate B cells, and these are the B cell which they are converted into plasma cells and eventually produce antibodies. They secrete antibodies. These antibodies are proteins which are specific to the protein present on the surface of the virus, so that these antibody they bind to the virus particle, so that virus particle is no longer free to cause infection, to infect the newer cell, and eventually this virus is eliminated by binding to the antibodies. So this is very important process in immunity, development of antibodies, and more important process in this part is development of memory cells. In fact, some T cells and B cells, they are converted into memory cells. Memory cells means these cells will remain in the body for longer time, and that time start from days to weeks to months and to decades also. So these memory cells basically they remain in the body, and when this same infection occurs again in future, this process of antibody development will not go in the same way. In fact, because if memory cells are there, these memory cells immediately will produce same antibodies as they have produced in the previous infection, and those antibodies will bind and neutralize the virus particle from the second infection also. So that's what happens in case of immunization. In fact, this process of immunization is the same. The purpose of any vaccine is to produce antibodies along with memory cells, so the memory cell remain for decades in the body. So next time when the virus comes again, it is quickly neutralized, because this process of natural neutralization take a longer time, first thing. Second, only antibody development is not the only product. When virus remains in the body, it keep replicating. That's how further complication also arise in the body. And sometimes when body is not able to cope up with the virus, this body surrender to the virus. So that's why it is not a healthy process to acquire immunity naturally. And to shortcut this process is called vaccination. So vaccines are basically nothing, but they are a mimic of the virus protein or virus itself, so that body feels like it's a virus, but actually it's not a virus. It's not a live virus. It cannot create any harm to the body. So that's what a vaccine does. So a vaccine is basically to train immune system, so that when infection occurs, this immune system is ready. It can secrete antibodies and neutralize virus within few hours, so that those complications because of actual virus infection what occurs, those are missing there. So that's why the vaccine is required. In fact, there are two different approaches to develop vaccines. One is classical platform. There are classical ways where people have developed many vaccines in the past, and then you have next generation platform. There are recent advancement due to recombinant DNA technology and biotechnology approaches. These methods, newer methods have emerged, which is employed to make vaccines. So in a classical platform, whole inactivated virus can be taken. Means a virus can be killed by using heat or any chemical agent, so that virus cannot replicate inside the cell. So that can be used as vaccine. For example, this Bharat Biotech, they have produced this first indigenously produced vaccine by India is Covaxin. So that is synthesized, that is produced using whole inactivated viruses, this approach which is classical, and in fact polio vaccine and other vaccines also have been developed using the same technique. Then you can use live attenuated virus. You can use same virus, but that has been attenuated. Attenuated means some genes have been removed inside, so it cannot replicate. It can only, it's a live virus, but it cannot produce more virus in the body. So that can be used. Then we know that antibodies are produced by the surface protein, which are present on the surface of virus. So those surface proteins can be used as vaccine also. So they can be given. They can be synthesized outside in the lab, and they can be used as vaccine. You call them subunit vaccine or protein subunit vaccine. Similarly, virus-like particles also can be used where they mimic only the outer surface of the virus. Inside there is nothing. Because we know that outer surface is important to develop immune response. Antibodies are formed against those proteins which are present on the surface only. So this also can be used. People have used it. And in next generation platforms, the first category is viral vector vaccines, where you use some virus which is harmless to the body, and you put genes encoding for surface protein into that virus. So when this virus goes and infect body cells, it does not harm the body. It cannot replicate inside the cell, but it can transfer the gene encoding for surface protein of coronavirus. So the cell start producing coronavirus protein, and then antibody response generated against those protein of coronavirus. In fact, Covishield and Sputnik V, they have been developed using this kind of approach. Then you can use DNA also, because we know that proteins are encoded by DNA. So you can take DNA fragment of the protein encoding for virus surface proteins, and then that can be injected into the body. So in body cytosol, DNA will be taken up inside the nucleus, and nucleus will transcribe into messenger RNA, then eventually protein will form. So the objective here in all the cases is to produce viral protein in the cell. Either you inject protein from outside, or you train cells, body cells, so they produce the viral protein themselves, which is used in order to develop the immune response. Or messenger RNA can be used. You can inject RNA directly, so that is translated into proteins, and those viral protein, they are immunogenic because they are foreign. Anything which is foreign to the body is immunogenic, that will be eliminated. Moderna and Pfizer vaccine in USA, they have been designed using this approach. Then you can mimic antigen presenting cell also, and although there is no example yet available, this is slightly complicated, but that also one of the approaches which can be applied in order to develop immune response and to produce memory cells. So next time when infection occurs, your body is ready to fight the battle and quickly eliminate the virus pathogen. Covaxin for SARS coronavirus is being developed by Bharat Biotech. In fact, Sinovac, Sinopharm, they also have used the same approach. So they have used the coronavirus, and we inactivated it using, you can inactivate using heat, but here some chemicals can be used. So those chemicals basically kill the virus. So only outer surface protein, they remain as such. Inside there is no RNA which can replicate. So this virus become harmless. So when you give this dose to the person, this virus goes inside, and because outer surface is intact, so it is taken up by the cells, it is processed, and immune response is generated against this, antibodies are formed. So it's a ghost virus, in fact, ghost vaccine also it's called, because the virus has been killed. So it's only mimicking the surface of the virus. Inside, its virus is harmless, it cannot replicate, it cannot create further complications in the body. That has been designed by Bharat Biotech, and it is available. Then Covishield and Sputnik V, these two have been developed using viral vectors. They are called viral vector vaccines. Here you can use a virus which is harmless to the body and can insert the gene encoding for the surface protein into DNA of that virus. So when this virus infects, it takes along the portion of DNA of SARS coronavirus also. So that is expressed in the protein in the cell, and eventually it generates antibodies. So this viral vector vaccine technology is based on integration of a foreign antigen. Antigen is the part of protein which bind, part of virus or protein which bind to the antibodies. So it takes that DNA encoding for surface glycoprotein and can be inserted into the vector of a virus DNA. So in fact here you can insert more than one type of antigen also. You can insert two proteins simultaneously, or even in the same vector you can introduce antigen for one disease and for another virus also. That also possible. But in case of Covishield and Sputnik V, this approach have been used, where in the viral vector genome one gene encoding for surface glycoprotein for coronavirus was inserted here. So in Covishield is developed by University of Oxford and AstraZeneca UK, and they have used a modified version of chimpanzee adenovirus. So adenovirus, they are harmless viruses. They are present in body also, human body as well they are present, but they don't create much harm to the body. That's why you can use this virus to transfer a foreign DNA along with it to the cells. So when this virus has been modified means some of the genes which are required for replication of the virus, they are not present in the virus. They have been removed by recombinant DNA technology methods, and instead in this DNA of this virus they have added a codon optimized sequence of spike glycoprotein. So SARS coronavirus is spike glycoprotein which was optimized. Optimized means because we know that the genetic code, the codons code for some amino acids. So initially coronavirus protein, they are good to produce protein for coronavirus, but they are optimized so that the same protein sequence is produced in human also. So after optimization, codon optimization, it was added into the genome of adenovirus, and then along with a sequence called TPA leader sequence. That's a tissue plasminogen activator leader sequence. This leader sequence basically guide protein outside the cytosol. So when this virus infects the body cell, it transfers DNA to the cytosol of the cell. And DNA contain a stretch of this coronavirus surface glycoprotein also. So in that way, the translation of this protein occurs in the cytosol. First the DNA is taken up in fact to the nucleus. In nucleus is converted into messenger RNA, and then in cytosol messenger RNA give rise to the surface glycoprotein of coronavirus. So this coronavirus protein has a TPA signal sequence. That means it doesn't remain in the cytosol, it is secreted outside, and that is required to develop immune response. That's why TPA leader sequence is added into the gene, so that this TPA leader sequence also expressed along with the protein. So when this enter into the cell synthesize this coronavirus proteins, and these protein which are synthesized by the cell, they work as antigen for the body, so that immune system become activated and antibodies are eventually produced by humoral response. Sputnik V also use same technique, but there is a difference that make it unique in the world so far. In fact, in first dose they use a different vector than the second dose, whereas in case of Covishield they use the same vector in both the first dose and the second dose, whereas in case of this Sputnik V they use one vector AD26, they call it AD26 to give primary dose, and second dose is given with a different vector, and they could see that using two different vectors the immune response was very high in comparison to the single vector vaccine. And when these vaccines similarly enter into the body, they produce the protein, surface glycoprotein of coronavirus, which is immunogenic, and immune response is developed. And the difference was observed in secondary immune response. So this is here when primary immunization was done. First dose was given of Sputnik V. The second dose was given here after 21 days, and they could see that there is a drastic difference. The secondary immune response was very high. It was very strong in comparison when only one vector was given as second dose. So that's why this is a unique technology where second dose, the booster dose, use different vector than the primary dose, and that aids in stronger secondary immune response production. Because it's not only the foreign protein, the coronavirus protein which is immunogenic, this vector also make it immunogenic itself. So if we summarize, all these three vaccines, they are safe to use. This is very important. They all are safe, and these efficacy varies. Efficiency of these vaccines, they varies. In fact, the Bharat Biotech produced Covaxin. It is known to have around 80% efficacy. It means if you give 200 people, 80% people they have got immunity against coronavirus. 20 people out of 100 they have chances to get coronavirus infection. Then Covishield is around 70%, and Sputnik V is over 90%. In fact, Sputnik V is among 3 vaccines which have more than 90% efficiency. So, but doesn't matter much. The efficiency doesn't matter much, because if you are vaccinating say 100% people in a population and your vaccine is just say 70% efficient, it means only 70% people are immunized, 30% people may get chance of infection. But if you have, if 70% population is already immunized, they have got immunity, then the 30% people will anyway, they have no chance to get immunity. That's what you call herd immunity. So the purpose here is, these numbers basically, I don't think it's wise to get away with these numbers, to think them seriously. Because if you immunize most of the people in a population, the rest of the people, they don't need to have immunity itself as per the herd immunity. So the objective here should be most of the people should get immunized. So when you have maximum people immunized, rest of the people who fall into say 30% who have taken Covishield, so they don't require to get immunity further, because of 70% people they are fine, they have got the immunity, so transferring infection will be minimized. So that's why in fact this is very important here to get vaccinated rather than indulging into which vaccine to be taken, because we also need to remember in India these 3 vaccines are given, they are not given to choose actually. We have a large population. One manufacturer is not enough to vaccinate everyone. So that's why these 3 are available in the market today. So I feel whichever vaccine is available, you need to get vaccinated, get it immunized immediately. In fact, I think even more partners should enter into the Indian market, so that quickly more and more people get vaccinated. The objective here should not be which is more efficient and which is less. The 70% or 80% efficiency is great enough for a good vaccine. So the idea should be quickly we need to get vaccinated, and most of the people should get vaccinated so that the further wave of coronavirus can be avoided. And that's the only way to stop the 3rd wave and 4th and so on. Vaccinate most of the people. So then whichever vaccine is available, one should get quickly vaccinated. Then the benefit outweigh the side effect of any. They have minimal side effects, these vaccines, and the benefit are so much, then those can be ignored easily. That's why all the vaccines, they have been approved after successful clinical trials. So all their effects, they are known to the scientific community, and that's why they are given in the market. So whichever vaccine is available, is idea to just take it up. So that's what is most important in today's time.